Alright, I'd like to begin this podcast by talking about calcium pumps. To begin with, I want to draw a cell here and remind you that calcium concentrations outside of the cell are high. They're about 1 to 2 millimolar. The calcium concentration in the cell is low, about 10 to the minus 4 millimolar, so a lot lower. Now, a few things about calcium I want to mention to you is that calcium is an important signaling molecule. It's involved in several pathways, including muscle contraction, fertilization, and neuron communication. So because of that, its concentration needs to be highly regulated. If this were a muscle fiber, we wouldn't want to have extremely high concentrations of calcium because we don't want those muscle fibers to constantly contract. So it needs to be kept at this minimum level. However, we want to make sure that there is a calcium store to use quickly. If you need to have a muscle contraction, it may not be advantageous to wait for it to be imported from the extracellular space. So what cells do is they have this ER, and the ER does lots of things, and we've talked about a few of those things, and we'll continue to talk about them. But one thing they do is they store calcium. Not quite as high as out here, but in that 0.8 to 1.0 millimolar range. So it's somewhere in between, but it's still quite a bit higher than what's in the cytosol. So how does the cell maintain a relatively low concentration of calcium in the cytosol while surrounded by a high concentration in the extracellular space and a high concentration in the ER? Well, not surprisingly, because we're talking about pumps, there's going to be a pump involved, both in the plasma membrane and the ER. Remember, we're going from a low concentration to a high concentration. So this is going to be an active transport pump, and it's going to require energy. And so what we have here is a calcium ATP driven pump, where calcium will move back to the extracellular space and calcium will move from the cytosol into the ER. This pump, let me say a few things about it real quick. It's similar structurally and the way it works as the sodium-potassium pump we've already talked about, except only one molecule moves. I should say one type of molecule. Sodium and potassium, remember, they exchange. One goes in one direction and the other goes in the opposite direction. The calcium only takes calcium and moves it this way. I should have wrote here, I better do it now so I don't forget. But remember, this is an ATPase, and so it requires ATP hydrolysis to move this calcium out. But it's an important investment of energy because we want to maintain a low concentration in here because we don't want this signaling molecule in the cytosol signaling various proteins if it's not time to do so. Another point I'd like to make about these calcium pumps is that by amino acid and nucleotide sequence, they are similar to the sodium-potassium pumps. The reason I mention that is because this suggests that the two of them have a common evolutionary origin. They both were probably derived from a similar protein pump or similar membrane protein that then was further enhanced through gene duplication probably to have multiple functions. Okay, now I want to talk about the reverse process. We don't always want calcium to be low here. Every now and then we want this store of calcium in the ER to go back into the cytosol. And we'll talk about muscle contraction later, but let's go ahead and at least mention it a little bit here. If this were a muscle fiber, it's a funny looking muscle fiber, but let's just pretend it is. If it receives a nerve impulse, so this is a neuron, as lovely as it looks, it delivers a nerve impulse where it signals the ER through various mechanisms, which we're not going to talk about now, to release calcium into the cytosol. In the cytosol, calcium binds this protein called troponin. This will activate troponin, and then troponin is an important protein that's necessary for muscle contraction. When we talk about the cytoskeleton, we'll come back to that later, but for now what I'd like you to know is that when muscle contraction is necessary, a neuron signals the muscle fiber, it sends a signal to the ER. The ER then releases calcium. Calcium binds to troponin. Troponin stimulates muscle contraction. Okay, that's all I want to talk about right now with the calcium pumps and the importance of calcium. But we'll come back to calcium when we talk about the cytoskeleton and when we talk about signal transduction. Alright, let's move on to osmosis. And as we introduce this topic to you, I want to remind you that simple diffusion, which we've already talked about, is the movement of solutes across a membrane. Going from a high concentration to a low concentration. Osmosis is similar, except with osmosis, it's the movement of water across a membrane. But like simple diffusion, it goes from a high concentration of water to a low concentration of water. Simple diffusion goes from a high concentration of solute to a low concentration of solute. So they're similar in that way. So if we draw one of these YouTube experiments, shown here, and if we want to talk about simple diffusion first, and we have let's first talk about simple diffusion, and we're going to place a permeable membrane here that allows the solutes to move back and forth. And if we're looking at simple diffusion here, we would expect solutes to move across this gradient from a high to a low concentration. So if we have lots of solutes, say on this side, and few over here, we would expect these solutes to move in this direction until we achieved some sort of equilibrium, where we had roughly the same amount of molecules on either side of the membrane. Now, osmosis at first is, I think, a little confusing because we're so used to thinking about the solutes moving, but we're going to think about the water molecules moving. And so we can use this same setup here to talk about osmosis, except now this membrane, which previously was permeable to the solutes, will now be not permeable to the solids. But water will be able to move across that membrane. We can think of this side here as having a high concentration of solutes, and this side over here as having a low concentration of solutes. But osmosis doesn't care about the solutes. It cares about the water. And so the concentration of water is going to be the opposite of the concentration of solutes. So this side here has a high concentration of solutes. It's going to have a low concentration of water. Over here, a high concentration of solutes means there's more water here. So here we're going to have a high concentration of water. If we allow for osmosis, the movement of the water, we will see it moving from a high concentration to a low concentration. That is, the water will move in this direction. So let's draw the results. Because it's the water movement from high concentration of water on this side to a low concentration of water on this side, our water level will go down. And over here, it will go up because we've had water move in this direction. The concentration of solute is going to stay the same on both sides because this membrane is still impermeable to the solutes, but not to water. So the water moves from high concentration of water to a low concentration of water. So now let's think about osmosis in a cell. Let's look at an animal cell here first. They're going to have this specialized channel here called aquaporin that allows for the movement of water. Animals are able to regulate how much water is inside of the cell fairly easily. And we don't get what we would call osmotic swelling. But we do see osmotic swelling, or we have the risk of osmotic swelling in cells like protozoans. And the way they eliminate their water that is coming inside of the cell at a fairly fast rate, and if it didn't eliminate this water, it would swell and burst. But what they have are these contractile vacuoles that will pump out water as fast as it's coming in. It's coming in pretty fast, but these contractile vacuoles will discharge the water from the amoeba. Plant cells can tolerate a decent amount of osmotic stress inside the cell. In fact, that's a lot of the reasons that you see that turgor within a cell of a plant. And they can resist this swelling because plant cells have a very thick cell wall. So they can tolerate some of this osmotic stress. But it is what helps those plant cells remain so rigid. Okay, I want to talk about transport proteins again. And I want to talk about how they can carry 1 or 2 solutes. They can do this with either active or passive transport. So let's draw our membranes like so. And now I want to spell out the 3 kinds of transport proteins that I want to talk about now. The first one is uniport. The second one will be symport. And then this last one will be antiport. Uniport will use this transport protein. As we've talked about before, it will bind to this transport protein and it will move through the membrane. Uni for one molecule being transported. The next one here is going to be symport. What happens with symport is you still have your 1 molecule or solute moving across the membrane, but we have a second molecule doing the same thing and going in the same direction. So sym is for these 2 molecules, solutes, going in the same direction. Sym for same. Then our last one here, we'll use these 2 same solutes, except they're going to move in opposite directions. Antiport. Anti for opposite. So the green one is going across the membrane from the top to the bottom. The orange is going from the bottom to the top across the membrane. Okay, now to kind of bring some of these ideas together, I want to talk about glucose transport. And to do so, I want to talk about the lining of our stomach. And to do so, let me draw these microvilli that aid in nutrient uptake. And there's going to be neighboring cells here that are going to be held together by these tight junctions we talked about. So we could, I'm not going to take this opportunity now, but we could review how proteins are blocked from moving across this area. But I really want to talk about transportation, since that's what this podcast is about. Now, in general, we have low glucose concentration here outside the cell in the gut lumen, and we also have low glucose concentration in the extracellular fluid down here. Our goal, though, is to get glucose from the gut down here so it can enter into our circulatory system. Oh, and I also forgot to mention that in the cell we have high glucose concentrations. So here we're going to have a symport transport protein where glucose, I'm just going to write it as a G, is going to bind here. as will sodium. Sodium will help drive this reaction, moving glucose from this low concentration to this high concentration. So with sodium, it comes inside the cell. Now remember, sodium is going to be something that gets pumped out of the cell using a sodium-potassium pump. So down here, we're going to have a sodium-potassium pump. That's going to take the sodium that's building up inside the cell because of this action with the glucose, and so it's going to be pumped out of the cell. At the same time, potassium is being pumped into the cell. In this way, we're keeping the concentration of sodium at its normal level. Again, remember, sodium's coming in, but the cell wants to keep that constant concentration of sodium, so it's got to pump it out. And it pumps it out by using an antiport system with potassium. So now we have high concentrations of glucose. Now we have to get rid of it because we don't want high concentration of glucose in the cell. But we really want to get it down here into the circulatory system so it can go to different tissues and provide energy. So we're going to go from a high to a low concentration. So in this case, we're going to be able to take advantage of a passive transport protein, where glucose will bind here, change the conformation, and then come back out over here. Okay, I want to run through this again quickly so we can review a lot of the important concepts from this podcast. So remember, the problem is we have low glucose here in the gut, and we want to get it to a low glucose environment on this side of the cell. The problem is that the concentration in the cell with glucose is relatively high compared to the gut and the circulatory system here. So it's got to move glucose from a low to a high concentration. That's going to require active transport. And it utilizes the sodium pump here and a symport mechanism to allow sodium and glucose to both go through this pump and into the cell. Now, to manage the amount of sodium that's in this cell, because we want to keep sodium at a constant level here, we need this sodium-potassium pump to pump out sodium in this direction and to pump potassium back into the cell. Glucose now is at a high concentration and it wants to go to this low concentration. So it's going to use passive transport, where it binds to this transport protein, changes the conformation, and releases glucose into this space here. Okay, I think that's a nice summary of what we've talked about. If you have any questions at all, please make sure you stop by and talk to me. If not, I'll see you in class. Bye.